Okay, so your introduction into genetics is going to look at millennial or millennial genetics, and that is considered the father of genetics because of his studies that he did with pea plants. So, a brief history is that people didn't understand that traits were inherited. They kind of saw that the traits between a child, some of them kind of look like mom and some of them kind of look like dad. And so the majority of the population before genetics, or the father of genetics, did his studies on pea plants, was that it was a blend of the people. So if you looked at a child and you saw part of mom and part of dad, with particular characteristics, nose, eyes, ears, then those particular characteristics were blended. And then you also had the particulate theory, in that traits are inherited as particles and offspring received different pieces. And so the piece of the nose or a piece of the ears, and we know those to be untrue. So Gregor Mendel was the father of genetics, and he studied pea plants. He found how particular traits were passing of the traits from generation to generation. And so Gregor Mendel looked at different characteristics with pea plants, such as the roundness of a pea pod or the color of a pea pod, if the plants were tall or if they were short. So he only looked at pea plants. The reason he did this is because pea plants are really easy to manipulate, and they produce generation and variations pretty quickly. And so you don't have to wait 9 months for a pea plant to produce peas, unlike a human or an elephant or other types of mammals that have long gestation times. Plants are pretty easy to manipulate and get to produce generations. And so different traits that Mendel was looking for were tall versus short, purple and white flowers, and then were the sounds wrinkled or were they round, were his main characteristics that he manipulated. And so as you can see here with this picture, you have different seed shapes, round versus wrinkled. You have flower colors, pod shapes, and pod colors. Were the pod shapes inflated like a balloon, or were they constricted like when a balloon shrinks down? Were the flower positions axil or terminal, and so those are just different describing words that we use when we talk about flowers. And then your stem height, or how tall the plant is it tall or short, were some of the traits that Gregor Mendel looked with pea plants. Another thing that's great about plants is we can fertilize plants. They don't have to fertilize themselves. We can take pollen from one plant and insert it into the female part of the other. So if you look here, you have the anther here with the pollen grains. We can take those and we can put them on the stamen, the female part of the flower, and then those pollen grains will go down and fertilize the ovaries of the plant. And so we can cross-pollinate plants. We don't have to wait on bees and butterflies and different pollinators to do that. Plus, we can control those experiments when we do it, and that's what Gregor Mendel did. So we can have true breeding plants, or we can have hybrids. And true breeding plants means that they always create the plants that look like themselves. So if you have a plant that is purple, it's always going to produce purple, or white, it will always produce white. So those are true breeding. And then we have hybrids, or an F1 generation. If you look here, it says F1, and so that would be like your next generation after you would cross the pollen with the plants for a purple and white. And so your hybrids are the offspring of the true breeding plants. And the reason we use true breeding plants is because there's no guess as to what their genetics are. So if they always produce purple, for many generations, like over and over and over again, you know it's purple. Same thing with the white. And so that when you get true breeding plants and you pollinate them, and if they, in this particular instance, produce all purple plants in an F1 generation, we call these hybrids, not in the meaning like it's a hybrid like a tiger and a lion coming together and producing a liger, a hybrid in a way of this is the generation or this is the population that's going to be produced from these true breeding white flowers. So that some of the traits are then dominant over others. And we talk about dominant and recessive in genetics as capital letters versus lowercase letters. Capital letters are going to dominate over the lowercase letters. So we also in genetics use capital and lowercase letters to demonstrate alleles. So when you start looking at different alphabet letters, and it doesn't matter what it is, when we're using the alphabet and we're using them in the expression of genetics and traits, we're going to use capital and lower letters, and they're specific to dominant versus recessive. So in this particular example, you have tall, which is going to be dominant over the short, and so the short is going to be a little t. And so that all tall offsprings are hybrids. And we should be able to see what this looks like in the next slide. Nope, I lied. Not the next one. So Mendel discovered that each trait is controlled by two factors, and those are the factors of an allele. So alleles are the letters that we use to express the traits. A through Z, doesn't matter which. And genes are the factors that determine your traits. So genes like you have brown hair, black hair, blonde hair, red hair. Those are the genes that determine which trait you have. And genes are located on our chromosomes. And we've talked about this, and that the genes on the DNA, our base pairs, A's, T's, C's, and G's, are going to determine proteins that are made. So the proteins that are synthesized. And when we're talking about genes, well, those are proteins that are synthesized. Let's skip over this one. So, Mendel's experiments. When we are going to do Mendel's experiments, we're going to look at Punnett squares, and we can call those monohybrid crosses. Monohybrid crosses look like this. Let me see if I can get something to pop up here for us. Punnett squares. And let me get an image up here. Here we go. That should work. Okay, look at this image down here. You have a Punnett square that has a yellow and a green. And so a yellow pod versus a green pod. Note that your big Y is going to be dominant over the little y. So in this particular example, you have big Y is yellow and little y is green. So your big Y and your little y's are going to come together in your Punnett square. Note that little y and little y is the only way you can get green, because if you put a big Y in it, let me see what this does. We'll go back this way. If you put a big Y with it, then what happens is that the big Y, or the dominant trait, will overpower the recessive trait, and so that you'll always get a yellow. Another example here is that you could have two big Y's. The two big Y's are always going to give you a yellow plant or a yellow pea pod in this example, and so it'll always be yellow. And so we have a few different ways that we can express the alleles. You can have two dominants and two recessives, or you can have a mix. So a dominant and a recessive, and we'll get to what those are called in just a minute. And so this is an example of a Punnett square that Gregor Mendel would have done with his pea plants. You can see that you cross these two, and then you can cross these two here, and then these here, and then these here. They kind of come out to be like a puzzle. And so you get 50% of your pea pods yellow, and you get 50% of your pea pods green. This carries for green, but that green is not expressed because the dominant, the uppercase Y, is going to control what trait is expressed. So, when a parent makes a sperm or egg, we call those that principle of making those particular types of genetics that can go in the eggs or the sperm. We talked about those as being a principle of segregation. And so there's no rhyme or reason why particular genes are going to show up in one egg or one sperm and not the other. That's also a process that we talked about crossing over. Remember the little pieces of the chromosomes that exchange genetic material that gives us all this genetic variability. And so when you have the principle of segregation, when you have all those different genes that are going to separate into your chromatid pieces that are going to go into the egg or the sperm, that gives us lots of genetic variability so that you get some of the genetic information from the mom and you get some of the genetic information from the dad. And so the diagram you're looking at here are parents. You have this parent as a short plant and this parent is a tall plant. And you do that cross and we get F1 generation. That means the offspring that are produced by these two parent plants. The offspring that are given to us by these two parent plants are both tall. And so note that you had a short and you had a tall, but both of the offspring are also tall. There are no short. Then we cross again for an F2 generation. And so with our F2 generation, we get 3 plants that are tall and 1 plant that is short. Okay, so parents, F1, F2, generation 1, generation 2. Note that we get variation, and this is where our short gene shows up, is in the F2 generation. So, with this particular diagram, you're looking at the different characteristics that Gregor Mendel studied with his pea plants. And then over here, we're looking at a cross, a different way to look at a cross or a Punnett square and looking at what goes into it. So we have a parent generation here. Notice you have big Y here, so you have 2 dominants. And you have little y here, so you have 2 recessives. And if those 2 cross, then down here, you're going to get more, but we're just going to pick out one for this, is going to be a big X, or a big Y and a little y as our F1 generation. So we're like, okay, well, we're going to cross this parent and this parent here. And if this parent and this parent cross, note you have big Y and little y for both parents in our F1 generation, you get this particular Punnett square. And so you get these different options. Note that there's a big Y here, a big Y and a little y here, and then 2 little y's. With this particular trait, we're looking at the round seed versus the wrinkled seed. And note you get 25% chance of having a wrinkled seed versus a 75% chance here of having a round seed. So, what does some of this terminology mean? Oops. We have genotype versus phenotype. Genotype is the genes that we're actually looking at, and we're using the alphabet to do that. So big T, big T, big T, little t, little t, little t are the letters that we're looking at. Phenotype is actually the physical characteristics. What is that going to look like? Is it going to be tall? Is it going to be short? Is it going to be wrinkled? So it's descriptive words, not the letters. The letters are the genotypes for what are the genes actually doing. And so over here you can see the genotype versus the phenotype. Tall, short is the phenotype. Big T, little t are the genotypes.